On the world's coral reefs, a small patterned shell hides a weapon that rewrites every rule of survival. This is no ordinary snail. It has a harpoon faster than the blink of an eye and venom so potent that a single drop can kill 20 adults. How did an unassuming mollusk become the ocean's most dangerous predator? The answer reveals why nothing about cone snails is normal and what that means for us. Across the world's tropical and subtropical oceans, a quiet presence moves beneath the sand and among coral rubble. Nearly 600 species of cone snails inhabit these waters, stretching from the reefs of the Indo-Pacific to the warm shallows of the Caribbean and the Red Sea. They live in places where sunlight filters through clear water, weaving patterns on the ocean floor. Some species settle in rocky crevices, others burrow under sand, and a few cling to the undersides of coral ledges, always just out of sight, rarely noticed by casual swimmers or divers. What draws human attention is not the animal itself, but the shell it leaves behind. Cone snail shells are small and tapered, rarely more than a few inches long, but each one is a work of natural art. Spirals of gold, white, chocolate, and crimson form intricate patterns, zigzags, bands, and dots, unique to each species. For centuries, these shells have attracted collectors, scientists, and beachcombers. In museum drawers and private cabinets, the glossy surfaces of Conus textile, Conus marmoreus, and Conus geographus shells stand as evidence of the ocean's creativity. Yet the shells serve a purpose beyond beauty. Their camouflage helps the snails blend into their environment, breaking up their outline against the shifting textures of sand and reef. Some species mimic the appearance of stones or coral fragments so closely that even experienced divers can overlook them entirely. Despite their striking appearance, cone snails remain some of the ocean's most secretive inhabitants. They move slowly, gliding over the substrate with a muscular foot, often leaving a faint trail in the sand. Most species are nocturnal, emerging under cover of darkness to avoid predators and the harsh glare of the midday sun. Their secretive habits and flawless camouflage mean that, for most people, cone snails are little more than a shell on the beach, a beautiful object to admire, pocket, or display. But behind the shell's delicate patterns lies a hidden world of adaptation and survival, waiting to be revealed. Hidden beneath the polished shell, the cone snail conceals a weapon that defies expectations, a harpoon engineered by nature with surgical precision. This harpoon is not bone or metal, but a single-use, hollow tooth built from chitin, stored deep inside the snail's body until the moment of attack. Each tooth is shaped like a miniature spear, complete with barbs at the tip and a channel running its length for venom delivery. The mechanics begin with the snail's proboscis, a flexible tube that can extend outwards, aiming like the barrel of a tiny cannon. At the tip, a ring of specialized epithelial cells forms a constriction, a biological latch that holds the tooth back. Inside, powerful muscles contract, building up hydrostatic pressure within the proboscis. The tooth is loaded, locked in place, and primed for launch. When prey comes within striking distance, the snail's nervous system triggers a rapid sequence. The latch releases in less than 1/10 of a second, unleashing the built-up pressure and firing the tooth forward like a bullet. High-speed footage shows the strike lasts just 1/10 of a second, a display of pure speed, faster than the blink of an eye. The barbed tip punches through fish scales or worm cuticle, injecting venom directly into the target's tissues. After the strike, the tooth's job is done. It cannot be reused. The snail discards it, often regurgitating the spent harpoon along with any leftover prey remains. Meanwhile, new teeth are already forming in the radular sac, ready to be loaded for the next hunt. This cycle of load, fire, discard ensures that each shot is as sharp and effective as the last. No other mollusk wields such a sophisticated projectile. The cone snail's harpoon system transforms a slow-moving animal into a predator capable of subduing swift, elusive prey. But the true lethality of this weapon comes from what travels through its hollow core, a chemical arsenal designed to overwhelm the nervous system in seconds. Inside every cone snail, the venom gland operates as a living chemistry lab, churning out a molecular arsenal so vast it challenges the imagination. Scientists have cataloged at least 50 distinct peptide toxins in a single species, and in some cases, the number climbs into the hundreds. Each of these peptides, known as conotoxins, is a miniature weapon evolved to target a particular lock in the nervous system of prey. One conotoxin might paralyze a fish by blocking its sodium channels, while another disables a worm by jamming its muscle receptors. The result is a pharmacological library, where every shelf holds a different solution to the problem of getting dinner in a crowded reef. No two cone snail species stock the same shelves. A fish hunter like Conus geographus loads its venom with peptides that shut down a fish's muscles in seconds, while a worm eater such as Conus textile produces a different blend, fine-tuned for invertebrate targets. Across the family, researchers estimate more than 50,000 unique conopeptides exist, each with its own sequence, structure, and effect. This diversity is not just a matter of numbers, it is a record of evolutionary trial and error, where every successful toxin leaves its mark in the genome. To make sense of this bewildering variety, biochemists group conotoxins into families, each defined by the type of ion channel or receptor it blocks. Alpha conotoxins target acetylcholine receptors. Mu conotoxins hit sodium channels. Omega conotoxins shut down calcium flow. These icons of the toxin world have become familiar to scientists, but the full catalog is still expanding. For prey, this means no two encounters are ever quite the same. For science, it means the cone snail venom gland is one of nature's most prolific sources of chemical novelty. A single strike from a cone snail is all it takes. The reason lies not just in the diversity of its venom, but in the way that venom is engineered and delivered. Within the snail's body, the venom gland stretches like a ribbon, divided into functional zones, each one a miniature chemical factory. These zones do not simply churn out toxins at random. Instead, each region specializes in a particular blend, allowing the snail to fine-tune its attack for different situations, whether it is hunting prey or defending itself from a threat. Every conotoxin begins as a simple genetic blueprint, but the real transformation happens after translation. Specialized enzymes add chemical modifications, hydroxyl groups, bromine atoms, glycosylation, even D-amino acids, altering the shape and function of each molecule. This post-translational customization turns basic peptides into a pharmacological arsenal. The result is a venom cocktail that can paralyze a fish in seconds or cause excruciating pain in a would-be predator, all depending on which zone of the gland is tapped. The potency of some conotoxins is almost beyond belief. In laboratory pain tests, certain peptides from cone snail venom have delivered pain relief at doses hundreds of times lower than morphine when both are injected directly into the spinal fluid. But the gland's true power comes from its compartmental control. The snail can selectively secrete from different regions, deploying a tailored mix for each encounter. This anatomical precision ensures that a single harpoon delivers exactly the right chemical punch, lethal, efficient, and nearly impossible to counter. Buried in sand or wedged between coral, the cone snail waits, motionless as the world passes by overhead. No chase, no pursuit, only patience. Its siphon, a slender tube, samples the water for the faintest trace of prey. Chemical cues drift through the current, the scent of a wounded fish, the subtle signature of a worm. The snail's nervous system interprets these signals with remarkable sensitivity, distinguishing not just the presence of food, but its distance, size, and even direction. A small fish, oblivious, strays too close. The snail's proboscis begins to extend, slow at first, then with purpose, uncoiling like a living periscope. The tip hovers just above the sand, tracking the fish's movements with uncanny precision. In a single fluid motion, the proboscis darts forward. The harpoon tooth, loaded and primed, shoots out with explosive speed, striking in less than 1/10 of a second. The barbed tip pierces scales and skin, delivering a precise dose of venom directly into the fish's bloodstream. Within seconds, the fish's muscles seize, neural signals short-circuit, and the struggle fades. The snail wastes no time. Its mouth opens wide, and the paralyzed fish is engulfed whole, head first, disappearing into the snail's body. Even prey 2 or 3 times the snail's own length can be swallowed in this way, the mouth stretching impossibly wide to accommodate the meal. The aftermath is silent. The snail retreats beneath the sand, digestion beginning as enzymes break down flesh and bone. This ambush method is nearly foolproof. Studies show that once a prey item is within reach, the cone snail's success rate approaches 100%. Slow metabolism means the snail can go for weeks, even months, before hunting again. In the world of reef predators, no other hunter combines such patience, precision, and lethality in a single package. 50 million years ago, the world's tropical reefs were already crowded with competition. In these bustling ecosystems, survival meant finding new ways to outsmart, outrun, or outfight the neighbors. Cone snails, once simple grazers, began to split into distinct lineages, each adapting to a different menu. Some specialized in worms, others in fellow mollusks, and a few, in time, set their sights on fish. This dietary divergence, scientists call them diet guilds, drove a remarkable evolutionary experiment. On the Indo-Pacific reefs, where biodiversity runs highest, the arms race between predator and prey became especially fierce. Fast-moving worms and alert fish forced cone snails to develop ever more precise and potent venoms, while prey species evolved defenses of their own. The result was a cascade of innovation. New venom genes, new toxin blends, and new hunting strategies, each tailored to a specific target. Conus textile, one of the vermivorous species, offers a window into this process. Its venom is a cocktail fine-tuned for subduing polycate worms packed with peptides that disrupt invertebrate nervous systems. The harpoon, too, reflects this specialization, stout and sharp, able to pierce tough worm cuticle. In the absence of natural predators, cone snails could afford to invest heavily in offense. Their shells became masterpieces of camouflage, not armor. The real defense was chemical, not physical. This predator release, freedom from being hunted themselves, allowed cone snails to pour evolutionary energy into refining their weapons. Over millions of years, the interplay of reef pressures and dietary specialization generated a family tree with more than 600 branches, each species a unique solution to the challenges of the reef. The venom arsenal of today's cone snails is the product of this relentless arms race, a living archive of evolutionary trial and error. It is this adaptability that now brings cone snails into contact with humans, collectors, scientists, and the curious, raising the stakes for both sides. A cone snail sting can unfold with terrifying speed. In the Indo-Pacific, where the world's deadliest species live, most human encounters happen by accident. Someone picks up a shell, not realizing the animal inside is alive and armed. The first sign is a sharp, burning pain at the sting site, often within seconds. Numbness spreads quickly, sometimes reaching the entire limb in minutes. Muscle weakness, difficulty speaking, and blurred vision may follow. For the most dangerous species like Conus geographus, paralysis can progress rapidly, threatening the muscles that control breathing. Without intervention, respiratory failure can occur in as little as 1 to 5 hours. There is no antivenom. Emergency treatment focuses on supporting breathing and circulation and often requires mechanical ventilation. Survival depends on how quickly the person receives medical care. Marine medics urge immediate action. Keep the victim as still as possible, apply a pressure immobilization bandage, and seek emergency help without delay. Even empty-looking shells can harbor a live snail. Never handle cone shells with bare hands. Use tongs or gloves and treat every specimen as potentially dangerous. In the late 20th century, Filipino scientist Baldomero Oliveira began collecting cone snails along the shores of the Philippines, drawn by a childhood fascination with their shells. What started as a quest for beauty soon became a discovery of molecules. In his university laboratory, Oliveira and his team isolated a single peptide from the venom of Conus magus, a fish-hunting cone snail found throughout the Indo-Pacific. That peptide was called omega-conotoxin MVYIA. It blocked a key calcium channel in nerve cells, halting pain signals at their source. For years, the molecule remained a scientific curiosity, too potent and too unstable for traditional medicine. But persistence paid off. By the early 2000s, pharmaceutical researchers had refined and synthesized the peptide, developing it into ziconotide. The drug was approved by the FDA in 2004 for patients suffering from chronic pain that no opioid could touch. Ziconotide is delivered directly into the spinal fluid, where its power can relieve agony in cancer and in nerve injury, offering hope where nothing else works. Yet the story is not finished. Ziconotide's benefits come with challenges, including high cost, difficult delivery, and the delicate balance of harnessing one of nature's deadliest molecules for healing. Every dose is a reminder that the line between poison and medicine can be vanishingly thin. Demand for cone snail shells has placed new pressures on these animals, especially in regions where rare species fetch high prices from collectors. The trade moves quietly, often slipping through gaps in regulation, but its impact can be seen in declining populations of some of the most strikingly patterned snails. For every shell displayed in a cabinet or sold at auction, there is a living animal removed from the reef, unable to reproduce or contribute to the ecosystem's balance. Cone snails depend on healthy coral reefs, yet those reefs are under siege from warming oceans, pollution, and destructive fishing practices. As coral cover shrinks, so too does the habitat available for cone snails and the countless other species that share their world. The loss is not only aesthetic or scientific, it is practical. Every cone snail species harbors a unique library of venom molecules, each one a potential tool for medicine or research. When a species vanishes, its chemical secrets vanish with it, closing the door on discoveries that could save human lives. Protecting cone snails means protecting the reefs themselves, and with them, the possibilities they hold for the future. Right now, scientists are racing to unlock new medicines from cone snail venom while their sting remains untreatable in the wild. Each discovery blurs the line between deadly and life-saving, reminding us nature's strangest designs often hold tomorrow's cures. The ocean's quietest predator is shaping the future of medicine, one toxin at a time. What else waits, hidden in plain sight?